My name is Dr. Chris Jenkins, and I am the CEO of the Ornate Society and the host of the Snake Talk podcast, the podcast where you learn about nature's most feared, maligned, and persecuted animals. I invite you to listen to this conversation, and maybe you'll find that what you perceive as fear is actually rooted in a deep fascination. Welcome to the Snake Talk podcast. I am here with Dr. James Emerson for round two. Hopefully all of you listened to episode number 122, where he talked to us about gray-banded king snakes and all kinds of interesting things about their ecology in the field. There's been very few studies of them, and he's conducted one of the few. So if you want to learn more about gray-banded king snakes, more about their ecology and studying them in the wild, again, go back and check out episode 122, and you can learn more about that. But what we're going to talk about today, and I wanted to get into it on that last episode, but we really didn't have time. So I just figured we would do another episode and focus in on rattlesnake ecology. As you all know, I am a huge rattlesnake fan, and I've seen a number of black-tailed rattlesnakes in the wild, and that's a species that James has done quite a bit of work with. So what we're going to do in this episode is kind of talk everything black-tailed rattlesnakes. We're going to talk, some general natural history, a little bit about their ecology, and then really focus in on James's research with the species. So welcome back to the podcast, James. How are you doing today?
I'm doing well. Thanks for having me back.
Yeah. So we're not going to go in and do the in-depth background and kind of trajectory in terms of how you got to where you are today. But I would encourage everybody to go back and listen to episode 122, and you'll hear a lot more about James's history and how he ended up, being a professor in Texas studying snakes. So, but for those who have not heard the last episode, why don't you just kind of give us the 30,000 foot level, kind of who you are, what you do, how you got into it, and where you're sitting today.
Yeah, for sure. So I am originally from the Texas Panhandle up in Amarillo, Texas. I spent the first large chunk of my life there. Did my undergraduate in wildlife biology up there at West Texas A&M University, then moved out to El Paso in the Chihuahuan Desert to do my PhD at UT El Paso, and that is where I did the bulk of my research on black-tailed rattlesnakes. And then after that, I worked as adjunct faculty at El Paso Community College until I eventually got the full-time position as an assistant professor of instruction and director of the undergraduate wildlife biology program at Texas State University here, where I'm technically teaching faculty, but I have managed to stay research active as well. So doing some rattlesnake research on timber rattlesnakes out here as well.
Excellent. Yeah, maybe we'll touch on that a little bit too. After we finish talking about blacktails. So we talked about the Trans-Pecos region. It's a region I've always been fascinated with. I've spent a fair amount of time in the region looking for snakes and just touring and doing a variety of things. And El Paso is obviously part of that. And that's where you spent a lot of time doing the research on some of these species. So we won't get into that region, but again, I encourage people, if they want to learn more about it, to listen to the past episode. But I do want to talk about black-tailed rattlesnakes, and we'll kind of start off by, we're learning we're learning a lot about rattlesnakes. We have a lot of, I don't know, a lot. We have a number of rattlesnake species that are kind of widespread species that vary across their range. And we're learning more about how those species, are they divided taxonomically within what we think is a species, either at a subspecies level, or is there more like phenotypic plasticity, and they just kind of look the same from one side of a country to 1,000 miles away, but genetically they're very similar. So let's start off by talking about what we know. What's the latest on black-tailed rattlesnakes, their distribution, and are they all one thing, or are biologists now breaking them out into different groups?
Yeah, so historically in the US, we had one species of black-tailed rattlesnake that fell under the umbrella of the northern black-tailed rattlesnake, Crotalus molossus. And then there are other black-tailed rattlesnake species down into Mexico in the Sierras down there and stuff like that. But some work done in 2012 kind of paved the way for my dissertation research at UT El Paso. And it was a master's student, Chris Anderson, in Dr. Eli Greenbaum's lab at UT El Paso. That in 2012, he published his master's work, which revalidated that we have two species of black-tailed rattlesnake in the United States. So we have an eastern form and a western form. And so the eastern form was revalidated as Crotalus ornatus because it was originally described that by Hallowell in the 1800s and then subsequently sunk back into Crotalus molossus. And so the initial hints of this were in some of Dr. Wolfgang Wooster's taxonomic research in the Central American rattlesnake group. And he was focusing on Central American rattlesnakes, but he used black-tailed rattlesnakes as his outgroup to serve as a basis for comparison. And with those limited analyses, you can look at his phylogenies and you can see that basically two groups of U.S. molossus kind of settled out of that. And so kind of the interesting thing from these projects is that even though these things have historically been considered one thing, those data showed that they weren't even sister species. So they didn't even share a most recent common ancestor. So those samples showed that Crotalus molossus was sister to Crotalus basiliscus, the Mexican West Coast rattlesnake, and that Crotalus ornatus was sister to Crotalus totonaccus, the Totonacan rattlesnake. And so the hypotheses surrounding that would be kind of a Crotalus molossus evolution along the western extent of the range geographically, and then eastward expansion towards the continental divide, which is now kind of recognized as the dividing line between the two species. And the eastern black-tailed rattlesnake evolved from eastern origins and then expanded westward towards the continental divide.
So I'm trying to think where the continental divide is as we get towards the Mexican border. Would that be over in New Mexico, I'm assuming?
Mm-hmm.
Kind of near the Chiricahuas, or?
It's kind of just east of the Boot Hill of New Mexico, if you're familiar with that. So
are those the Floridas, the mountains?
The Floridas are a little east of there, but you get like kind of Hatchet Mountains. You move I'm blanking on the names of kind of those mountain ranges kind of north of I-10, but there's also the Hatchets are south of I-10, kind of just just east of the Paloncillos there. But you get kind of those sporadic mountain ranges as you're driving west on I-10 or east on I-10 if you're coming from Arizona or California.
Mhm.
You see the signs for the Continental Divide, but it's the major uplift there where the drop of water that falls kind of west
Mhm.
of the Continental Divide siphons off to the
Yeah.
west, and east
Is it
siphons.
near? I wonder. Is it near Lordsburg? Trying to
Yeah,
think.
kind of in
Yeah.
that area. Like
Yeah. I
I
think.
think you pass the sign on I-10 just east of Lordsburg.
Gotcha.
Um, and so, but
And
then, oh,
so,
sorry.
do these two species, as we're talking about the Continental Divide, do they come in contact in that region of the Continental Divide, or are they separated by a physical barrier today?
Yeah, so they are in theory sympatric along those Continental Divide mountain ranges. I've talked to a bunch of people that have spent a lot more time in those specific Divide mountain ranges than I have. And evidently, you can come up with some weird looking things. You have things that look like, you know, um, textbook Melosis, things that look like textbook Ornatus. And then you have like these weird looking things, even though kind of visual differences are pretty limited between the two and somewhat inconsistent. Um, and so there are a lot of people that are pushing for um, new research to be done focusing on that Continental Divide region so we can see like levels of gene exchange and hybridization and that kind of stuff, see what all is going on there. And then also, what would be nice is if we could get an increased body of samples from Mexico populations to have more confidence what's going on with the black-tailed rattlesnake south of the border.
Mm-hmm. Gotcha. So, what is the common name of the species east of the Continental Divide? Is that would people call it the ornate black-tailed rattlesnake, or just the ornate rattlesnake?
So I've heard it called the eastern black-tailed rattlesnake and the ornate black-tailed rattlesnake kind of interchangeably, because my initial meetings with those authors on the 2012 paper, I have always like deferred to calling it eastern black-tailed rattlesnake. But I think if you look at like SSAR common names and stuff like that, I think they accept ornate black-tailed rattlesnake.
Gotcha.
Um,
Okay.
but I know my publications, I referred to it commonly as eastern black-tailed rattlesnake.
Yeah. And I've seen them across, their range in the US, which means I've seen both species in the wild. And it strikes, but I don't have a lot of experience working with them like you have. But it strikes me that maybe relative to some of the other species, especially in the Southwest, that maybe they have a little bit more general habitat requirements, distribution, and correct me if I'm wrong, but how, in just a very general sense, within their distribution in the U.S., what types of areas do you find them in, or are they kind of across a variety of different habitats?
I would consider them to primarily be montane generalists.
Mm-hmm.
And so they tend to be found in rocky mountainous regions, but within those regions, they can be found from relatively low elevations all the way up to extremely high elevations. They can inhabit a wide variety of habitats within those as well. And so I think I think it's pretty accurate to say that they are um, montane specialists, but within those montane ecosystems, they are generalists.
Okay, that makes sense. That's interesting because there's obviously a lot of montane specialists in that region, and a number of them are more specialized in that kind of micro habitat within the broader range. So that is interesting. Let's kind of go in and talk about your research specifically. We might kind of spin off on tangents and talk about different aspects of their ecology or natural history, but I'm assuming this well, remind me, but I think this is when you were in El Paso, but I may have that wrong. But talk a little bit about kind of how you ended up getting interested in black-tailed rattlesnakes and studying their ecology, and kind of what was the beginning of all that.
Yeah, so really, I was interested in kind of more specialized, the smaller montane rattlesnakes. As I started talking about doing my PhD. And so it was with talks through my PhD advisor, Dr. Jerry Johnson at UT El Paso, that I focused on black-tailed rattlesnakes. And so I chose to do my PhD at UT El Paso because they had the amazing resource of the Indio Mountains Research Station, which we talked about at length last time. Um, but with this revalidation, Dr. Johnson was basically like, you can do Lepidus stuff if you want to, but we've already had PhD students work on Lepidus here. And so you don't have a blank canvas, so to speak. He was like, with this recent revalidation of black-tailed rattlesnakes, you basically have a blank canvas to do whatever you want with them. And so, kind of after orienting myself to the research station and getting familiar with some of the Trans-Pecos snake species, I was like, oh, this seems like a really amazing species to work with. And so, kind of made that choice and hit the ground running to help fill in these gaps on what Crotalus ornatus does in Texas. I know some researchers in the past had done small scale telemetry on them, but none of that information was publicly available. So there's no literature basis for things like home range size, movement frequencies, movement distances, movement windows, all of that kind of stuff. In terms of publicly available information was very scant.
Mm-hmm. So you're going to use radiotelemetry. So you're going to study the animal's movements. Maybe there's other components to the study, whether it be habitat or thermal. But how did you design it? Meaning, like, kind of how many animals were you planning on tracking? You've already talked about they kind of have a wide berth in some areas in terms of like elevation gradients. Where you focus in certain types of elevations or other environments. Were you studying all males, all females? How did you design the telemetry study?
Hey everybody, I just wanted to take a quick break and tell you about an urgent issue facing one of the most incredible tortoises on the planet. The Seychelles Archipelago, a crown jewel of the Indian Ocean, is home to breathtaking natural beauty and unparalleled biodiversity, but a looming threat now endangers one of its most iconic species, the Aldabra giant tortoise. A new luxury development on neighboring Assumption Island threatens to unravel centuries of conservation work, putting these gentle giants and many other species at grave risk. You can make a difference. A global movement is rising in defense of Aldabra's fragile ecosystem, and you can stand with conservationists fighting to protect one of the last great sanctuaries of the giant tortoise. Visit www.friendsofaldabra.org to learn more and sign the petition to make your voice heard today.
Yeah, so we wanted, for one chapter of my dissertation, to be just a general description of their behavioral ecology. So space use, movement patterns, habitat use, microhabitat use, and those kinds of things. And so kind of how we just studied it is I randomly select 1 to 3 kilometer transects that went through a variety of habitat types, and I would hike those on a given night, trying to look for any snakes that I could find, because if we were going to incorporate those habitat analyses, we didn't want sampling to be biased, because if you look for black-tailed rattlesnakes where you can find black-tailed rattlesnakes, that's not really informative on habitat selection versus use, that kind of stuff. And so that's how I sampled snakes. We were looking for generally equal representation of males and females, which would eventually, those females would become important to a different chapter in my dissertation that I'm sure we'll get to later. But yeah, so we wanted generally equal sample sizes. We wanted kind of reproductive adult classes of snakes. So during the breeding season, we can get those mating behavior observations and stuff like that.
Okay, great. Well, let's talk a little bit about what you learned with that, relative to that first chapter. Meaning, sounds like fairly, really what you're looking at here is a new species that hasn't really been studied, and you're trying to almost describe movement patterns for the first time. So just talk to us a little bit about what you learned from the telemetry in terms of snake movements and all the components within that.
Yeah, so from a movement perspective, I mean, we kind of were able to confirm, at least at the research station, that movements seem to be pretty standard for desert-dwelling, monsoonal climate rattlesnake species. And so by that, I mean that movements tend to be fairly infrequent in the dry parts of the year, and then that monsoon season hits in late to early July into early August through late August into mid-September, and then movements take off. And so that also coincided with their breeding season out there. So they seem to have a single fall annual breeding season, which once again is very similar to a lot of other montane desert dwelling rattlesnakes. And during this time, males basically have no boundaries as to how far that they will move to chase females. And so some of my large males would move a kilometer in one movement bout, chasing males over multiple ridge lines in the mountains. And so pretty impressive movements for something that's normally considered fairly sedentary, like a rattlesnake.
But for much of the year, throughout those dry seasons, it sounds like they're really not moving that much. Are they, when they're in that more sedentary mode in the drier parts of the year, are there certain types of places that the snakes are living in, or did you find them across a variety of different habitats? And did you have a sense for what they were doing at that time of year? I'm assuming they were foraging, but did you have a sense for what they were doing then?
Yeah, so most of my snakes during those kind of lower movement times of year, they would be either in these highly fissured, exposed bedrock areas. So we categorize those habitats as rocky slopes. And so they'd have a lot of deep, presumably high humidity, cooler temperature crevices that they could retreat to. But also kind of an interesting thing to me, when they were out, they seemed to really like succulent vegetation. And so like those thick opuntia groves, prickly pear groves, and yuccas seem to really be a critical microhabitat component for them. And I didn't do any analyses of the characteristics of those specific places. But I would assume that with the water storage ability of succulents, if you get up under those plants, there's going to be kind of a humidity bubble that reduces evaporative water loss and kind of buffers you from the heat.
Yeah, I wonder what that might do to prey. Do you think it would? I mean, there's obviously prickly pears are fed on by some things, but do you think some of the prey species would also kind of focus in on those little humidity bubbles?
For sure. We do see our kangaroo rat mounds and our Catadipus burrows, our pocket mice burrows underneath those succulents as well. And of course, our wood rats will build middens in the hearts of those prickly pear clusters because they get some degree of protection from the spines.
Mm-hmm. So these snakes are spending a fair amount of their year during the dry times kind of in different habitats, but kind of in a real micro sense, oftentimes associated with these succulents, probably for kind of the physical environment, possibly for the prey environment as you're talking about it. And then these monsoon seasons hit that coincide loosely with breeding season. And so, but then everything kind of goes out the window with that. And these snakes really start moving long distances. I'm assuming maybe not kind of irrespective of habitat. They're kind of, they're looking for females. And so they end up wherever those females might be. Is that an accurate assumption?
Yes, I believe so. And interestingly enough, one of the subtleties that I forgot to mention was that those super long monsoonal movements were exclusively males. And so females, it seemed like they were more hunkering down and restricting movements in those monsoon seasons. And I think it's so the males could find them. And so I actually saw, it wasn't statistically significant, but I saw a slight kind of just visual peak in the movement data for females in June, which is one of the hottest, driest months. And so my hypothesis would be that those females are going to move around despite the unfavorable conditions because they want to lay down those pheromone trails. So once the monsoon hits, those males can find them.
Mm-hmm. What are the some of the life history characteristics? I don't know if you. It sounds like it's not a long-term study, so you're probably not able to estimate those. But do we know much about life history characteristics with this species, meaning things such as, like, with females, like ages to sexual maturity, pregnancy intervals? Are they having, are they having babies every year, every 2 years, every 4 years? Those types of things. What do we know about the kind of that life history traits of this species?
Yeah, so not a whole lot. I think just based on Mark recapture data on the research station, we kind of have an idea that they probably have similar lifespans to other kind of medium to large rattlesnakes. And so there are individuals that have been recaptured that, were at least 12 years old, but we don't know exactly how far that pushes into, some of the estimated ranges of 30 plus years, whether or not that's true or not with them. But the most I ever tracked a single female was for 2 consecutive years. And in those cases, they did not reproduce consecutively. And so my guess is that the Indio Mountains are in multiple rain shadows, one from a large mountain range to the south in Mexico, and another from a large mountain range to the north in the US, the Eagle Mountains. And so they are relatively low and dry. And so given the amount of resources that a female rattlesnake puts into reproduction, my guess would be that it would probably be maximum once every 3 years.
Mm-hmm.
And that's if, she's doing great. And this kind of ties into another chapter that I did, but I did just anecdotally, once again, sample size for my reproductive females was, I think I had 5 gravid females, so pretty small in the big scheme of things. So all of this is just anecdotal. But I kind of saw like what seems to be 2 broad strategies based on body size. And it seems like the big females that I had, so 800 plus grams, I think the biggest female that I had was like 950 grams, something like that. But they seem to give birth to more smaller young. And so they would give birth to 5 to 7 neonates that were, 20 to 24, 25 grams. Whereas the smaller females seem to give birth to fewer, larger babies. And so the smallest female that I had, I didn't even know that she was gravid, and kind of an interesting, because she wasn't a transmitted snake initially. And so the interesting thing about finding her was that I was tracking one of my large males, and he had returned back to this cavity underneath a rock. Look back there, there was a small female in there with him. And so I was like, oh, this is great. They probably just mated because it was mid-August breeding season. And so I was like, we'll go throw a transmitter in her and I can follow her around. But we processed her in the evening. The next day, we were going to do transmitter surgery on her. We went into the lab where she was housed, and overnight she had given birth to 2 giant babies. And she initially weighed, I believe it was like 198 grams the night before. After she gave birth to these 2 babies, her body mass immediately dropped to less than 100 grams. I can't remember. She is either 96 or 98 grams.
Wow.
And one of her babies was 34 grams and the other was 36 grams. And so she basically lost like 55% of her body mass with just those 2 babies. And it seemed to be a pretty general pattern. I had a couple more smaller snakes, and they gave birth to 2 and 3 offspring as well, but they were all upper 20s to low 30s in grams. And all of those females seem to lose, 45 to like 55% of their body mass. Whereas those bigger females, they would lose about the same amount of weight. So they would lose about 100 grams after giving birth, but, is proportionally a much lower percentage of their pre-birth body mass.
Yeah. Interesting. So in studying the female component here, as you mentioned, they kind of hunker down and you were speculating a little bit about what that might be relative to the breeding system. But it sounds like you were following both gravid and non-gravid females. And as I'm sure in many rattlesnake species, it's been documented where, your non-gravid females are out feeding, acting much more like, say, a foraging male or any other foraging snake, whereas your whereas your gravid females are moving relatively shorter distances, finding certain features, often rocks, that provide just like the perfect thermal environment to maintain kind of constant body temperature day and night. Did you see differences in gravid and non-gravid females relative to movements and habitat use and all of that?
Yeah, absolutely. And I'm actually trying to revisit those data now because I kind of want to put out an kind of in situ summary of reproduction in the species based on those data I have from my dissertation. But essentially, what I saw is that my gravid females would not move. Like once they came out of hibernation, brumation, the in the winter, they would choose a crevice or a rock that they were comfortable with. And it was usually pretty high up a southwest facing slope in a very hot microclimate.
Mm-hmm.
And they would basically just be up there directly basking, and they would only move, a foot or two depending on their thermal needs. And then a few days before they would actually give birth, they would actually move across the top of whatever ridge they were, incubating those babies on. And they would move to kind of the north or northeast facing slope and find a nice shaded, humid crevice and give birth in those.
And then they would do kind of parental care, stand with the young, but in that new, more humid environment. Is that right?
Yeah, so I really only observed this with one of my gravid females because another chapter in my dissertation required that I collect gravid females and have them give birth in captivity so I could do some feeding trials for innate prey preference on them.
Mm-hmm.
But the one instance where I didn't get the female and she gave birth in a crevice, she was very defensive of those babies. And so I was able to count 8 in the crevice with her. She chose kind of this like 3-foot tall, like vertical crevice that kind of opened towards the base and had a cavity that went maybe 2 meters back into the bedrock.
Mm-hmm.
And when I found her, she was coiled at the front of that expanded opening at the bottom of that vertical crevice. And so I tried to be sneaky and reach over her with my tongs so I could, just temporarily collect as many of those young as possible, just so I could get like morphometrics on them and stuff like that to summarize the litter. Because since they were born in the wild, I couldn't do any innate prey preference stuff because they've been able to smell those cues for however short a period of time.
Mm-hmm.
But as I tried to reach over her with my tongs, she would and this was a very steep, rocky slope that was a bit treacherous to be on, but she would bull rush me every time. Like the tongs went over her head, she was coming out after me. And then I'd pull the tongs back, she would pull back into her coiled position. And after trying for a couple of times, I was like, okay, she's a good mother. I've stressed her out enough. I'm going to leave her be, and I'm not going to fall and break my arm or something like that.
Well, that's a good transition. Talk a little bit about what we know about diet with these ornate blacktails, and then what sounds like you at least had a lab component to preference, but just anything you can talk about relative to feeding, diet, anything you learned in that realm.
Hey everybody, I just wanted to take a quick break and tell you that the Ornate Society is dedicated to protecting some of North America's most at-risk species and their habitats, like eastern indigo snakes in the longleaf savannas, eastern hellbenders in the Appalachian highlands, and wood turtles in the great northern forests. Healthy ecosystems depend on these incredible animals, but these landscapes and species need our help to survive. If you care about reptiles, amphibians, and the wild places they call home, visit www.orien.org to learn more about our work and how you can get involved.
Yeah, so most of our knowledge on the diet comes from examining stomach contents of museum specimens. We also have some anecdotal evidence based on people that have tried to keep them in captivity as well. And so from those stomach contents, it seems to be, kind of the standard suite of appropriately sized native desert vertebrates. And so most of the diet is small mammals, whether it's kangaroo rats or pocket mice, depending on size of the individual, or there's also quite a few birds that are represented in the diet. And then when you get down to young snakes, occasionally some lizards like greater earless lizards and stuff like that show up, just because for a small neonate rattlesnake, a lizard is a relatively safe, appropriately shaped food item that's ubiquitous in the desert and easy to consume. So that's fairly intuitive. In terms of keeping them in captivity, a lot of people have tried it, and they are kind of attractive, large rattlesnake species, but they don't make very good captives because a lot of them seem to be hesitant to accept, your typical white lab raised house mouse that most keepers are going to have the best access to feed their captive snakes. And so they seem to be a little less generalized in that capacity when compared to something sympatric like a western diamondback rattlesnake, or even a rock rattlesnake, which is a lizard specialist in the wild, but they readily accept appropriately sized, house mice in captivity. And so,
I have a rock rattlesnake, and that thing loves, pinky rats. I mean, from day one. Anyways, go ahead.
Yeah, and I've kept some wild caught rock rattlesnakes as well that presumably were eating mostly lizards out there, and they have no hesitation to take a house mouse, a frozen thawed house mouse in captivity. But with the blacktails, it's a very different story. And so most people that keep them, they end up having to cruise around looking for road-killed kangaroo rats or other native small mammals that they can either freeze and then directly feed, or they can use them as tools to scent those lab mice in order to trick them into eating something that they don't really want to. And so in order to kind of determine whether or not like prey preference was innate or this was something that they learned over time, I did an experiment with 10 neonates that I was able to get from my snakes. And I guess I will say that one of those gravid females that I got those neonates from, she was not a transmitted snake, but she was actually out in what appeared to be ambush one night, and that's how I found her. So I think she may, despite being very, very gravid at the time, I think she may have been hungry and trying to find a meal when I initially saw her. And so what I would do is I would have a big open top sterile arena, and I'd put the snakes in there for a day just to let them settle down, so they weren't as kind of freaked out by the new surroundings. And I went out and I trapped a bunch of whatever small mammals I could on the property, and I gave them a 10-minute bath in water, and then I froze those water samples until 10 milliliter sample sizes, and then I would use those to present the scent cues to the snakes. And so I only did species that I was able to catch 2 or more individuals of. So you have a variety of individual representation in those water samples. And then most of my samples were small mammals, but I did have greater earless lizards, Cophosaurus texanus. I also, just because they were super common and I could get samples of them, I was like, oh, this is a common potential food item. So let's see how they feel about red-spotted toads. And then I also, because rock rattlesnakes have been documented eating a lot of centipedes, I also took samples from some big Scolopendra centipedes to present to them as well. It didn't have the federal permit, so unfortunately I didn't do any bird sampling to get scent cues, but that would be a really interesting twist on it. Mm-hmm. But yeah, so I sat them in these, once they had acclimated for 24 hours in these arenas, I had 2 blocks of wood. Each of them had a paper towel on them, and randomly would select 1 block of wood to put just a tap water control on. And then the other block of wood would blindly be selected for random scent cue. And so, and then, when you do the tap water, both of the blocks have the tap water control out there. And then you'd leave them alone and record their behavior for 2 hours and see if they, how long it takes them to encounter the scent cue. When they encounter it, how much tongue flicking do you see? And so you'd actually count the tongue flicks, and then ultimately the decider for if they really enjoyed something was if they assumed a stereotypical ambush posture facing the scent cue, and then how long they remained in that ambush posture. And so you would take all these things and calculate tongue flick ambush score, which is called TFAM, and then you would run those through the comparative mix models to determine if any of the TFAM scores were higher for specific scent cues over others, so on and so forth.
So what did you find there?
Yeah, so not surprisingly, I found that appropriately sized native small mammals were the preference. And so since these were neonates, overwhelmingly the strongest feeding response was to Catodipus intermedius, which is the rock pocket mouse, which are these tiny little heteromyid rodents that are very common in the Chihuahuan Desert. Secondarily, they, despite being neonates, they really liked Merriam's kangaroo rat scents as well. So I presume as they grow into adulthood, that's going to be their favorite prey item. And there were also there was also a minor peak for greater earless lizards as well. And also, I will say that my initial trials were kind of in diurnal situations. So I had them with, just standard lights. So you could get good videos of this stuff. But when I saw kind of this lower peak of earless lizards, I decided that when I was radio tracking these animals, at the research station, I would find them in an ambush during the day, at night, all sorts of whatever time they were out. If they found some place that seemed good for food, they would set up ambush. So I decided to repeat the trials nocturnally and just to see what happened. And there was no difference between diurnal and nocturnal behavior. But
Interesting. Well, are there any other kind of components from your dissertation that speak to the ecology of these ornatus? And if so, what were those?
Yeah, and so I guess before I transition to that, one other thing that I forgot to mention with the innate prey preference trials was that the very first litters that I collected, they had been fed on lab mice for 2 years by the time that I got around to running the trials. And the later litters that I collected for around a year. And despite having only been fed these things in captivity, they really did not like the house mice scent cues. And so I think, if I remember correctly, I had 0 ambush postures assumed on house mice, even though that's what they were familiar with eating in captivity. And so that kind of goes to strengthen that anecdotal point that I made earlier, that these things are pretty hardwired for these native small mammals over just any broad spectrum small mammal scent cue.
Yeah, interesting.
But yeah, and so as you're doing telemetry, as you know, one of the coolest things about telemetry is you stumble upon like unique observations and behaviors that you might only see once, and so you can't do any real robust quantification of those behaviors, but they're interesting to use to generate hypotheses and stuff like that. And so one of those was the fact that I found that small female that I mentioned earlier that lost so much weight with those 2 babies, with the male coming back to her, the day before she gave birth. And so I always think, is he like coming back to assist with like neonate attendance or not? And, there are a few other researchers that I've just talked about in passing that see similar things. And so it's just to me, it's an interesting thing about, because the more we look at rattlesnakes, the more we see these really fine-tuned, ingrained behaviors around parental care and stuff like that. And so it makes me wonder if there are cases where, biparental care occurs in rattlesnakes, and what drives that, and how common is it, those sorts of things.
Yeah. Interesting. Well, so no other takeaways from your dissertation work that you can think of?
There's some. It's just, to me, for an endothermic snake species, it just always amazes me how adapted they are to that desert habitat. Like, I would find, like in May and June, like the hottest parts of the year, I would find snakes up in vegetation when like ground temperatures were like 74 degrees Celsius, which is insane.
Yeah.
I forget the exact conversion, but it's something like 160 degrees Fahrenheit on the surface temps down there. And so they would just move up into yuccas to get off the hot ground and stuff like that. And also you see the things that you always hear other researchers talk about faithful use of different landmarks within their home ranges. And so I had one snake that he would have a very short overwintering period every year. So I think one year he overwintered for like 6 weeks. The other is like 7.5 or 8 weeks. He would basically go into his crevice in mid-December and come out in, early February and be going about his business. But every time, both times, I say every time, but both times that he came out from hibernation throughout the tracking period, he would always visit like the same prickly pear cluster, like right off the bat during the season. And then he would always move to the same feeding areas and stuff like that. So I think.
And so that was a shorter duration in hibernation than the other snakes.
Yeah. So more typical would be like early December, some late November, they would go into hibernation and they would stay there until early March.
Okay.
And out there, I don't think that hibernation is primarily driven, or brumation is primarily driven by, temperature, because you get, it gets to be 80 degrees during the day in December and January, a lot of times. And so I think it's primarily moisture availability, since the winters and spring are very, very dry. If they're active during the day, they're going to be subject to a lot of evaporative water loss, and temperatures in the desert cool off rapidly at night in the winter. And so they can't really be nocturnal because of that temperature restriction.
Okay. Well, great. I'm glad that you shared all of that, because it's like I said, it's like a newly discovered species, and being one of the first people to study their ecology. I mean, obviously, there's probably similarities, to the Western version, but it is fascinating. So, before we begin to wrap up here, though, I would love to just hear. I'm just talking the real, like, short 2-minute version of, like, you talked about working on timber rattlesnakes now that you've, kind of relocated within Texas, and maybe just talk about, what you're, planning, what you're kind of starting to examine with timber rattlesnakes, and the direction you see that going in the future.
Yeah, for sure. So the current research that I'm working on is a telemetry based project on timber rattlesnakes. I am working on a private land area, which is a little over 10,000 acres. It is owned and managed by the Cynthia and George Mitchell Foundation, who they do a lot of stewardship and sustainability work with private landowners in Texas. And so they're kind of an important, nonprofit organization here, which I know is near and dear to your heart. And so at this property, we have some molecular data on what is going on with the population. And so I got this position here, and so I paired with the faculty member, Dr. Forstner, who has those molecular data, and we're trying to kind of figure out an all-encompassing, kind of population biology project for what is going on this property. There's a lot of conservation implications with timber rattlesnakes in Texas, as not too long ago, their protections were downgraded at the state level. And where they are found is also highly developing habitats. So they're facing rapid urbanization, especially in Southeast Texas with the sprawl of the Houston Metroplex and stuff like that. And so trying to figure out what populations are doing in order to inform future conservation and management issues. We got through field season 1. We're starting field season 2. One of the big things we're interested in is what their overwintering behavior is. So are they denning solo, as seems to be the case with many of these southern timber rattlesnake populations, or are they communally denning like some of the more eastern, northern kind of rocky habitat populations? And so this year we're also going to hopefully incorporate SFD monitoring protocols, because the population hasn't been tested for SFD, so we don't know if it's present, we don't know if it's not. But trying to get ahead of that, if it is present, is important to us. And then also just another techniques thing that we're trying to do with this. I know you had her as a previous guest, but Dr. Waldron her lab pioneered the epoxy and tie rattle attachment method for radio transmitters. So in order to, since we know that timber rattlesnakes are pretty stress prone, if you disturb them a lot, they are known to vacate important ecological areas for them. We are, trying our hand at this transmitter attachment method. So hopefully it can be less invasive and we can make kind of have more informed interpretations about behavior based on these telemetry data that we get. And so far through 1 year, we've had great results with it.
Yeah. Well, interesting. I'm looking forward to see those results as they start to come out. So we've been going about an hour here, so I think we're going to wrap this one up. But before we do, I would love to hear your second best snake story, because we just heard one in the recent episode.
Yeah, and so for the last one, since the theme was gray-banded king snakes, I tried to keep it on topic with the life of a gray-banded king snake. For this one, earlier in 2024, I actually had a note come out summarizing this. So if anybody wants to see kind of the full detailed view of this story, it is in volume 1 that came out in March of 2024 of this year's HERP review. But we'll go back to the desert, and this involves an encounter with a Sonoran lyre snake, Trimorphodon lambda. And so I was in the Chiricahua Mountains, and I saw a lambda crawl across the road. And personally, Trimorphodon in the U.S. is one of those fascinating species to me, because you look at the bulk of their distribution and it's subtropical to tropical, but in the U.S. they're a desert species, a desert genus. And so, of course, if I see a Trimorphodon, I'm going to try to, get pictures of it. This one was crawling across the road. I got out and picked it up, and I noticed a stream of headlights heading my way on the road. And so right as I noticed that, she latched on to my forearm. And every other Trimorphodon I had encountered, they will kind of do these mock strikes where they hit you with their mouth, but they don't actually latch on and set those rear fangs in you and like try to envenomate you. And so, so as she latched on, I was concerned with moving my vehicle off the road and getting to kind of a safe place to, take a GPS point for what I found, all that kind of stuff. And so it ended up that she chewed on me for nearly 1 minute. And, everything I've heard about trimorphodon envenomation, regardless of the species across their range, was that it's relatively insignificant to humans. So, just maybe a prolonged like trickling of blood, some local pain, stuff like that, but nothing to be super concerned about. And so I was like, okay, gently kind of worked her back fangs loose, let her go, continued looking for herps in the Chiricahua Mountains. I noticed as I was continuing to look around, the pain in my forearm started expanding and got to be kind of like this weird, like radiating numbness and then like shooting nerve impulses. And so it's kind of like a really weird pain. And then like 3 hours after, I began to get violently sick. And so it started off with like sharp stabbing pains in my stomach. And then I actually started like vomiting and stuff like that. And so I was like 3 hours away from home. So I was like, I got to get back to El Paso. Like, this is not good. And so like the whole trip back, I'm driving down the border highway that goes through western El Paso, through southern New Mexico into southeastern Arizona. And normally there's a high border patrol presence on that road. So I'm stopping like every 10 to 15 minutes to throw up on the side of the road. And so I was just like, these guys are going to think that I'm out here and I've been like drinking or doing drugs, and they're going to try to like take me in or something. But fortunately I didn't. This was like the only time I've driven on that road that I didn't encounter border patrol. And, but yeah, I got home to El Paso shortly after I got home, like the puking subsided. And so, cause I was like kind of on the fence, like, should I go into the emergency room or not? And, but then the puking subsided. And for like the next few hours after that, it just felt like I had like stabbing abdominal pains. And, ultimately that went away in another few hours. And then, but my forearm was like swollen and bruised for like nearly 2 weeks after the fact. And so, I kind of like, this is kind of an embarrassing story to tell, but it also, I think it's important because people that are herping and are going out catching these things, sometimes they get a little cavalier with the rear fang species because they tend to not be dangerous. But it just highlights that anytime something is injecting like proteins and enzymes into you, the potential is there for a bad time. So maybe be a little bit careful and don't let them chew on you if they do bite you.
Yeah. And so were there other records, in writing this note, did you encounter other records of similar bites with lyre snakes, or?
Yeah. So as far as I can tell, this is the first kind of evidence for systemic symptoms of envenomation from a trimorphodon. All other accounts of envenomation are very localized symptoms. And so, yeah, it was interesting from that standpoint. And I even like at conferences and stuff, I talked to an individual who did a lot of postdoctoral research on opisthoglyph venom compositions in Mexico and Central America. And he was like, yeah, nobody's ever told me a story like that. But then he was breaking down like the components of their venom that probably caused the symptoms. So he's like, it makes sense that like the metalloproteinases would cause the nausea and stuff like that, so on and so forth.
Yeah. Okay. Well, very interesting story. I encourage everybody to go check out Herp Review and read the account. And just wanted to thank you again for coming on the podcast for round 2. So I enjoyed the conversation.
Yeah, thanks for having me, Chris.
Okay, great. I just wanted to thank the audience and tell everybody to remember, snakes are animals too, and it's a privilege to see one in the wild.